Computer turned on. All right, we're ready. So having no remembrance, because God knows class was like 5 days ago, and in the pandemic that seems like it may as well have been like a year in some ways. But we were talking about neuromuscular structure. We were talking about sarcomeres. We were talking about how we have these actin filaments and these myosin filaments, and those two proteinaceous filaments are the things that will interact with each other and give us force generation, which is going to lead to all movement. So I think we talked about some of the myosin side of things, but on the actin side, I'm pretty sure I mentioned that every actin molecule is a little ball or a little sphere, okay? And you're going to get individual actin molecules that are going to get kind of lined up like this, and then they're going to have two strands of those that are going to twist around each other, and you're going to get an actin molecule. On every actin molecule, there is a site where myosin wants to bind. Myosin wants to grab onto and connect to the actin, okay? That is myosin's sort of one never-ending seek-it-out job. It wants to grab onto actin and therefore form a cross bridge and generate force. So in order to keep us from just being one giant muscle cramp all the time, we have to have this regulatory protein called tropomyosin, which looks like a rope, okay? It's going to cover the length of the actin filament, and it sits down in and covers up the myosin binding site on the actin. So you've got your filament, the myosin's trying to bind, but it can't lock in, okay? At various points along the actin molecule, we're going to have these other regulatory proteins called troponin. And troponin has multiple subunits. The one we're really concerned about is troponin C, but the troponin is going to connect to the tropomyosin. And the troponin C subunit will bind calcium. Specifically, it binds calcium that has likely been released from the sarcoplasmic reticulum, okay? So we're going to get our signals all the way down into the muscle. We're going to release calcium. It's going to bind to the troponin. That's going to cause what's called a conformational change, okay? So a change in shape of troponin. So as the troponin rotates and it changes its shape, it is connected to the tropomyosin. So when it moves, it pulls the tropomyosin away from the actin, from the binding site, okay? That is going to open up the myosin binding site on the actin and allow myosin to grab on, okay? Then we're going to kick the calcium out, away from the troponin, okay? It's going to get taken back up into the sarcoplasmic reticulum. Then the troponin is going to flip back to its regular place, and it's going to push the tropomyosin back over the myosin binding site, okay? So in this way, we can go from normal state, no muscle contraction, no force production whatsoever, to, in theory, when we want it to get turned on, we'll release calcium. That will open it up and allow us to form a cross bridge, which we're going to talk about next. Okay? Does that part make sense? Okay? Very good. Okay. There you go. And well, that part works. It doesn't want to advance. There we go. All right. So everything that we've done up until now, everything we've talked about is to get us to this point, okay? This right here, okay? The formation of a cross bridge when myosin binds onto actin, this is the whole game, okay? All of exercise physiology depends upon this. Everything that we do, this, okay? You want your heart to beat, this. You want to move, this. You want to expend more energy, this, okay? This is the one thing. Myosin binds onto actin. When they are bound together, we call that a cross bridge. Once they are bound, then the myosin, because it is a molecular motor, will attempt, it is not always successful, but it will attempt to pull, okay, to generate a force and pull the actin in a certain direction, okay? We will talk about next week why sometimes it can pull and why sometimes it is trying to, but it is woefully unsuccessful, okay? Sort of be like me if I tried to push Alex and I couldn't make him move. I'm getting lots of cross bridges and I'm trying to shorten them, but they're not going anywhere, okay? This is the whole ballgame. When the head tilts and pulls on the actin, we call that a power stroke, okay? The power stroke yields force. It yields movement, right? So in this one little thing, everything else we're going to talk about for the rest of class is in many ways traced back to and wrapped up in this one particular interaction, okay? Okay, now, I think I told you guys that there are some other proteins that are in a sarcomere. We focus mostly on actin and myosin. Myosin is roughly 2/3 of all of the protein in your muscles, okay? But there are a few other ones, and I want to point a couple of them out because you may encounter them as you read papers, okay? Nebulin, okay? Alpha-actinin, desmin, these are Z-line proteins. Nebulin is a bit of a structural protein within the thin filament. Alpha-actinin and desmin are going to be anchoring proteins on the Z-line. These are going to function as like a scaffold on which we will build out these thin and thick filaments and anchor them. The last one on here, we could probably do 2 weeks on all by itself now, okay? But I don't have those 2 weeks to spare. And it's a giant protein called titin, okay? Originally, titin was thought of as this elastic structural protein that occurs and kind of anchors filaments at the Z-line, okay? What we now know is that, one, it does do that, okay? But in most of your undergraduate exercise phys books, they don't say this, but what we now know is that titin is also a molecular motor. Titin can and does generate its own force independent of myosin, okay? At one of our national conferences a few years ago, I was able to see in real time electron micrograph pictures or videos of titin generating force, okay? I assume at some points from a guy at the University of Toronto, his name was Walter Herzog, I assume that at some point the guy will probably win a Nobel Prize for that, okay? Because for forever we thought myosin was the muscle contracting molecular motor. We now know that titin also contributes to some of this. It changes the entire scope of everything, okay? So helps thick filaments center between in there, but it also generates some force, especially when muscles are being stretched, especially at very long muscular links. There's some thoughts about how we could maybe do some specific training to increase titin above and beyond some other things. This is relatively new-ish stuff for us. We don't have a lot of data yet to know for sure how much this contributes to force. We know it does, but it's hard for us in a whole muscle to kind of get at that. So you may see some things about this, okay? All right. That's the muscle side, all right? Now we're going to move. Now we're going to switch over and we're going to talk about the nervous system side, okay? Pearson, stand up. Have you ever played the game Simon Says? Back up a little bit, we'll get you away from Alex. All right. Have you ever played the game Simon Says? I have. You have, okay. Well, today it is more like Dr. Black says, okay? I want you to do something for me. Okay. I want you to just raise your right hand out in front of you and wave at me. Now put it down, okay? Now I want you to take your left leg and go out sideways, okay? Put that down, okay? So I ask him to do something, he doesn't, right? voluntary control of skeletal muscle. Doing those movements required him to contract his muscles, right? That starts in the brain. Okay, we're not going to get into all of the kind of real sort of stuff going on there, but in general, he has thought. We're going to get into the motor cortex. We're going to generate action potentials. It's going to get sent down through the brainstem, down the spinal cord, and eventually out to your muscles to contract. We're going to get that side of stuff. We're not going to worry about the brain stuff too much. Okay. Now, for my next trick, what I want you to do, Pearson, is this. Okay. Can you not breathe for 10 seconds? Okay. 5, 6, 7, 8, 9, 10. Okay. We can watch your mask. You have one of the little ones on, which is partially why I picked you. It was easy to see if you're breathing from doing those things. So he's not breathing. Okay. Some level of voluntary control over at least not contracting a muscle there. Make your heart stop beating for 10 seconds. Can you do that one?
I don't believe so.
You don't think so?
Well, probably eventually, but
do you want to try?
I don't think we should.
We probably shouldn't. I mean, 10 seconds isn't going to be like the worst thing in the world. We got some AED machines outside. We can bring it. Thank you, Kristen. You may sit down. Okay. Why can't he stop his heart from beating? Yet he can stop breathing. He can do whatever he wants, right? If I tell him within reason, like wave at me, pick an arm up, pick a leg up. What's different about cardiac muscle versus skeletal muscle? It has autorhythmicity, which is a big fancy term for it beats on its own. Okay? One of the things that makes skeletal muscle somewhat unique from, say, smooth muscle and cardiac muscle in your body is that it is under voluntary control. So that means there's a connection from the brain, okay? The central nervous system connects out through the peripheral nervous system to skeletal muscle, which gives us the ability within some, kind of constraints of we control on and off. And what's going to really blow your mind is when I tell you that that's all that neurons can do. They're either on or they're off, right? So how on earth then do we figure out I need to generate 1 pound of force versus I need to generate 100 pounds of force out of the same muscle when all I can do is it's on or it's off, okay? We're going to get into that. It's kind of interesting. That's one of the unique things, okay, about skeletal muscle. Yes, Meg?
So what about like when you're sitting there and like does that?
I mean, there's a whole host of reasons as to why that might happen. Maybe it's not as random as you think it is. Anybody ever had a cramp? Okay, that's a muscle contraction that you probably didn't want to happen, right? Okay, so the problem with all of this is there's a huge number of steps from brain all the way out to formation of cross bridge. And so if you get an erroneous signal where it gets activated somewhere along this big long chain, when you haven't told it to from motor cortex, you may get a muscle contraction. Okay? You have a cramp, you're trying to make it stop, and it's not stopping, right? That's a very kind of muscle specific, we would call it a peripheral issue, we think. Okay? We don't really know. I mean, we can tell you what's happening when you have a cramp from a physiology sample. We don't really know why you have them in some instances other than they're associated with being dehydrated and having some muscle damage, and I assure you it has nothing to do with eating bananas or not. But that's kind of all that we really don't know. Okay? So that's why you may get those things. Now there are some other instances. There's some very small but rare instances when you can get skeletal muscle contractions outside of cramps and twitches and things without there being voluntary control. And they're called reflexes. Okay? And so there may be some issues. Well, what you can see here is we're going to go from brain down the spinal cord, and then in the spinal cord, there are specialized neurons or nerve cells called alpha motor neurons And for some reflexes, some signal out here in the periphery is going to get transmitted back to the spinal cord, and it's going to make the alpha motor neuron depolarize and get a muscle contraction, typically to remove you from some sort of harmful stimuli most of the time, okay? You touch something hot and you pull away before you even actually really know that it's hot, okay? It's meant to prevent tissue damage. Now, I could sit up here and it's hard to do it without, I got to get really relaxed, but I can, the tendon tap reflex, right? We're stretching the muscle, we're making use of the muscle spindles and the 1A afferent and those kinds of things, but some sort of peripheral input, an afferent signal that bypasses all of this, because the worry is that it has to go all the way up and all the way back down. That may take too long, and there may be serious like tissue damage. So we need to do that, okay? Or the other instance with some of the like the stretch reflex stuff is like I'm running or I'm jumping and I'm up here in midair, and all of a sudden I've changed my position from what I thought I wanted it to be, and so then I need to be able to adjust force production and stuff without my brain is watching the basketball, and I need to adjust what's going on down here, and so there's this kind of, we don't want to overtax the brain. But we'll get into a little bit of that stuff later on, okay? All right. Okay. So, as I said, the nervous system, once we kind of go brain, spinal cord, in the spinal cord there are these neurons, these we call them alpha motor neurons, okay? This is the midpoint or the way station for how we're going to transmit these signals, generally in the nervous system, okay? Not exclusively, but generally, as it relates to muscle contractions, we're going to signal using action potentials, using electricity. This is good because it goes really fast, and we have quite a bit of distance we may need to cover. What you're going to see is there are about 250 million muscle fibers in an average person. We have about 450,000 motor neurons, right? So what you're going to have to guess and take from all of that is that obviously each motor neuron is going to have to connect to more than one muscle fiber, okay? But every muscle fiber, almost all of the time, almost every muscle fiber is only going to be connected to one alpha motor neuron, okay? So what we call that, or we term that then, is each motor neuron and the individual fibers that it connects to or that it innervates, that's termed a motor unit, right? When we are going to get into next week talking about force production, okay? How the nervous system turns on the muscles to generate force and then controls how much force we get, the motor unit, okay, is going to be the basic functional unit of the nervous system, right? We're going to do things one alpha motor neuron at a time to try to get some precise amount of force, okay? Are we okay with that? Okay. Some muscles, okay, have thousands of motor neurons. Some muscles have a much smaller number of alpha motor neurons. Some motor units are very, very small. You got the alpha motor neuron and 10 or 20 muscle fibers. Other ones are going to be the alpha motor neuron and maybe thousands of muscle fibers. Okay? And again, all of that is done for a reason that is going to help impart function to that muscle based upon the job that needs to be done from that muscle. The motor units and the number of motor neurons that go to the muscles in your eyes are going to be somewhat different than the ones that go to your glutes. The ones that go to your hands are going to be different than the ones that go to your quads or your bicep, because those body segments have different jobs and different functions, okay? Things like your fingers, where we need very close, kind of fine motor control, may have more motor neurons that are very, very small. So we can very precisely control how much movement we're getting and how much force we're getting. Whereas things like your glutes or your quads, okay, your gastroc, big muscles, strong muscles, right? You don't write using your quad. Quad's job is squat, right? So for those, maybe you don't need as many. Our force gradations may be a little bit smaller and don't need to be quite so small. And the goal is just generate lots of force, control the muscle in that way. Okay, so it's going to be very task specific based on what those muscles are going to do. Again, structure is connected to function. Okay? Even though it's all muscle, and the muscle's all the same, even though all the alpha motor neurons are basically the same. Okay? But we can change kind of how these things are organized ever so slightly, and that will give us slightly different functions. So motor neurons are all in the spinal cord. The motor neurons, all of which go to one muscle, so all the motor neurons to my bicep, let's just say, okay? They're all going to be in the same place, roughly, in my spinal cord. So at like C5, that's where my bicep, and most people are right around C5 is where your bicep motor neurons are all going to be located, okay? In general, cervical vertebrae, upper body muscles, okay? Thoracic vertebrae, a little bit of stuff in here, right? We're going to get down then into the lumbar and very, very high sacral, lower body muscles. The only exception, diaphragm. Diaphragm's way up at like C3, C4, okay? Way up here, those neurons are better protected, right? Less stress put on the spinal cord and stuff all the way up here, unlike down there, because your diaphragm is so important for you to be able to breathe. That's an odd sort of, doesn't make a lot of sense, kind of evolutionarily, or at least the first person that had that, it wouldn't have made a lot of sense evolutionarily, but it's turned out to be pretty good, okay? There is usefulness to this and a downside to this. The good is that by all of those motor neurons being in a very similar place, if signal starts in the brain and it works its way down, then what this tells us is that if I need to turn on all of the motor units in my bicep or my tricep, or just pick a muscle, then there's not going to be much time delay from the signal from my brain being able to arrive at all of those motor neurons. Okay, so imagine if I had some bicep ones here, and I had some bicep ones at say T10, and I had some bicep ones down at L3. And so what happens is that, well, these ones up here at the top, we're going to fire first, then these are going to fire, then these are going to fire. Even though it's electricity, and that time delay may be just fractions of a second, that's going to have an impact on how smooth the contraction is, how much force we can generate, et cetera, et cetera. Okay? What's the downside? Are you okay? Okay. What's the downside? Kelvin? Why is it bad to have them all in the same place?
Not sure.
Okay. Megan, would you like to try to help him?
So if you had a spine injury in a certain area, then it's like
All done. That's the downside. If there's an injury to the spinal cord, or there's an injury to a particular area in the spinal cord, either everything below that level, it's all gone, or if it's you get just like it's an incomplete injury in a particular area, it may wipe out all of the neurons for a particular muscle. So like my arm may work fine, but I have no bicep or something, okay? And so there's some interesting things that go around that. For anybody that cares, if you want to do physical therapy or anything like all of that, you know, neurology will tell us that if somebody has inflammation in and around, they've got an injury, if you've ever like slept funny, if you haven't yet, you will give it time. You're young, okay? You'll be like me. You'll sleep funny, and you'll wake up, and you will have this shooting, crazy ass pain in your neck that runs down here in your deltoid, down from your bicep, right down, right, the part here to your thumb, and you go to the doctor, and they're like, oh, well, look at that. I guarantee you what you did was you squished something at C5, because biceps is weird, it's tingly, it's weak. The inside here, right down to your thumb, those are all motor neurons that are at C5, right? Tricep is fine. Why? Because it's at C7. Doesn't matter. Okay? So you can learn, these are called, sometimes they call them dermatomes, but you can diagnose where people have impingement or low back injuries or different, like, or they have some kind of impairment and stuff, simply by looking at what muscles work versus which ones don't work. We can trace our way back to the spinal cord and try to figure out what's happening. Okay? Okay. So we did this. Here is, let's just say, a single vertebrae, okay? This is one vertebrae. We'll just pick something. This is, I don't know, it's L3. So maybe these are like something where the quads are going to be, okay? You're going to have, right, what's called a dorsal and a ventral side, right? Kelvin's heard this, but so I've never had anatomy, which is probably an astounding thing to you guys. Maybe it's not. I've never had class in anatomy. I've taught anatomy and physiology, never had anatomy. So dorsal, ventral, I kind of get my stuff messed up. I always think about sharks, like the dorsal fin. So this is the back of this, where the person's back is. This is sort of relatively towards the front, okay? But in and around, in one kind of slice of vertebrae, there's going to be what we call a ventral horn and a dorsal horn, and there's going to be nerve cells or neurons that are going to be interspersed all in there, okay? On the ventral side, here, on the inside portion, that's where there's going to be the alpha motor neurons, okay? All of their collective axons then are going to project out through the foraminal space in these, what we would call these big spinal nerves, okay? On the dorsal side, there's going to be a collection of other neurons, but these are going to collect sensory information from roughly the same place in the body where these are going. So if these are going to the quad, this is going to be afferent information coming in. And then within the spinal cord, and we'll talk about this a little bit later, maybe probably not today, but within all of this, these neurons talk to each other, okay? Through either direct interactions or through interactions with these other neurons called interneurons, which are just kind of like intermediaries that are going to be there, okay? And this lets us then kind of control what's going out, what's coming back in, and we can make all of these kind of fine adjustments. When you change position or something weird happens, what's going to be there, okay? Okay, more things about those. Probably, maybe everyone but Pearson has seen what a neuron, kind of this basic structure of what a neuron looks like before. Yes, no, Alex, maybe. Okay, Megan's nodding her head at me like, please, God, let's not do this again. So that's okay. So neurons or alpha motor neurons consist of what we think of as the neuron. So this cell body that's in this part's in the spinal cord, coming off around that cell body of these little like whiskers, if you will, on a cat. Okay, they're called dendrites. Their job is to gather information from surrounding neurons, the surrounding environment. They're going to pick up information and then bring it in here, okay, to the cell body. Projecting out from the spinal cord, out through the foraminal space, are going to be these axons. When most people think of nerves in your body that you can see, what they are thinking of are bundles of axons, okay? I was a baseball player. We're always thinking about that the pitchers mess up their ulnar collateral ligament, which are ulnar collateral nerve, right? They tear their ulnar collateral, ulnar collateral, there's an ulnar collateral ligament and an ulnar collateral nerve. And it's here, but you could open somebody up, you can see the nerve. That nerve is going to be made up of all of the axons that are coming from the spinal cord that are going down to there. They're also going to have the 1A afferent axons that are coming back up. But axons come out, leave the spinal cord, they're going to go out and essentially connect to the muscle tissue. So spinal cord, axon leaves, runs all the way down, okay, to wherever that muscle is going to be. Axons can be very, very long. And So because of that, we got to move a signal very fast. Axons, most motor axons are going to be pretty big, and they're going to be surrounded by this fatty tissue called myelin. We're not going to do the electrochemistry of why larger axons are going to move signals faster, why having the myelin on here is going to help us move signal faster. All I want you guys to know is these axons have myelin. They tend to be pretty big in diameter, and that means that the signal moves fast, which is a good thing. There are some pathological conditions where this myelin goes away and your movement slows down. Okay? All right, so that's those things. We've talked about this, muscle bodies, and this stuff is just kind of basic stuff to look over. Dendrites, motor axon. All right. Last bit of here that I want to really get at is going to be a very specific structure at the end, where the axons are going to come into very close contact with the individual muscle fibers. We're going to call this a synapse or the neuromuscular junction. It's sometimes called the motor end plate. That's an old term. You don't see that very, very often anymore. Okay? For the most part, it's called the neuromuscular junction. Okay? Axon comes out. At the junction, there is no physical contact between the nervous system side or the end of the axon and the sarcolemma. No physical contact. Okay? Don't ask me why. I don't freaking know. It doesn't make a lot of sense, but no contact. Okay? How about these kinds of things? So a lot of this other stuff that is in here, there's the stuff called the axon hillock, there's microtubules and neurofilaments. One of the issues with axons, and one of the issues with the nervous system is that the cells are in the spinal cord, and sometimes the neuromuscular junction end of things are way out, like down in your feet, okay? And so the nucleus is in here, and it has this network of filaments where we're going to make proteins and neurotransmitters and things, and we've got to ship them and move them all the way down the axon. Okay, so there's a lot of kind of interest in researching how all of that stuff works and those things. But I just want to kind of present it as this is a thing that happens. Okay, myelin, Schwann cells. Okay, motor units. We talked about these a little bit. So here is here's what I think is a really cool image of neuromuscular junctions. Okay, so here we have individual muscle fiber, individual muscle fiber, individual muscle fiber. Here are the ends of the axon, right? So here's our big kind of collection of all of them. This is what you would see as a nerve. The axons are going to begin to kind of branch off, and they're going to come down, and then they're going to have this very highly specialized structure on at various kind of places in and along these muscle fibers. Okay, so you've got one here, you've got one there. Okay, so we're going to look now a little bit more closely at what's going on at the junction itself. Nope, we're not going to do that yet. We're not going to do that yet. We've talked about motor units. Okay, so there's an extra slide there. My apologies. So here's a cartoon of what I just showed you in an actual microscope picture. Okay, what you can see here, this is the nervous system end. This is the sarcolemma or the muscle side. Note how the structure at the junction of each of those on each side is a little bit different. On the nervous system side, we've gone from kind of this nice, what you think of as kind of a regular kind of thin tube, and then it bulbs out at the end, like a light bulb, okay? We call this a bouton, which I always think is a hilarious sounding word, but that's the bouton, all right? On this end, okay, on the muscle side, there are going to be these folds in the sarcolemma that are going to be there, okay? Now, why do you guys think? Why do we think we need these special structures, that we need the change in the structure, okay? When you get past all of this, the sarcolemma is just straight and flat. For most of the part, the axon is this little narrow structure, but you get down to here and it bulbs out. What's the advantage of having both of these things? increases surface area. Okay? It increases surface area. So on the nervous system side, there's more area to have more acetylcholine in the end here, which is our neurotransmitter, to be able to then be released so that we can make sure that we're going to transmit our signal onto the muscle. On the muscle side, by having these folds, there's more surface area so we can have more receptors per kind of unit area there for acetylcholine. Okay? And so what this is going to do is it's going to ensure as good and efficient a transmission of a signal from the nervous system side onto the muscle side as we can possibly get. Okay? Because, as I told you guys, it takes a long time. Okay? Imagine you're a tiny little, there are other molecular motor proteins, there's things called dynin and kinesin. You're a walking protein, and you have to traverse from somebody's, poor. Alex, how tall are you? 6'4". 6'4". So jealous. It's got to go, like 4 feet from Alex's spinal cord all the way down, and you're a thing that's like 10 microns across. So that's like running, a super long, like 50 miles or something to you. So to make some parts of acetylcholine up in the spinal cord and then move that shit all the way down to here, it's going to take a long time. So once we've expended all that time and effort to do that, what we don't want to do is to get our action potential to here, release the acetylcholine, and then have it not actually bind onto the receptor and do what we want. Also, by doing it this way, if we release just a little bit of acetylcholine, we've got a really good chance, right, that it will bind and actually do what we want it to do. So we don't have to have this huge, vast extra supply. It's going to be on that. Okay? So it comes off, it's going to bind. Once the acetylcholine has bound, it is going to then, there's an enzyme that is going to be in the basement membrane here on the muscle side that's going to actually break the acetylcholine back off of its receptor. That enzyme is called acetylcholinesterase. Okay, so what happens is it binds, we activate these receptors, they generate an action potential in the muscle. Okay, it moves along the sarcolemma, and voila, from there we're going to get some stuff going on the muscle side of things. Then the acetylcholinesterase breaks the acetylcholine off. It's going to float back up, and a lot of it will then get reabsorbed back onto, back up into the bouton. Okay? Again, we don't want to have to keep making it and bringing it down from the spinal cord because that will take forever. And your poor little kinin, kinesin, and dynin will just be very tired. And that costs us energy. Those things have to move, so it takes energy to move them down. Okay? Okay, so this area right here is called the primary cleft. The areas where there are the folds in the sarcolemma are called the secondary cleft. We have lots and lots of acetylcholine receptors in both the primary and the secondary cleft. We did these, basement membrane, boom, okay. So there's some interesting things about acetylcholinesterase and about acetylcholine. There's a host of neurotoxins and a host of chemical weapons that do one of two things to those. They either cause massive release of acetylcholine, so you get this sort of completely on signal, right? Or what they do is they prevent acetylcholinesterase from working. There's a very famous thing called sarin nerve gas. Listening to a book about the Iraq War, and they were talking about that Saddam had used sarin nerve gas when it's ever really been used in like real life. He used it on some folks that are called the Kurds that live in North Iraq, I believe. Anyway, you release sarin nerve gas and it blocks the acetylcholinesterase, and so you get this constant activation there, and it won't ever let go. And as you guys will see later on in class, that leads to fatigue. That happens in your diaphragm. You can't relax your diaphragm, so you can't breathe anymore, and you suffocate. Okay, so it's really terrible stuff. Although before you get to that point, like every muscle in your body has contracted all at the same time. It's very, very terrible. There's some other ones though that prevent the release of acetylcholine, that prevent the binding of acetylcholine, something called tetrodotoxin, which I think is in pufferfish. If anybody likes sushi, you can get pufferfish. If you go to like Japan or South Korea or someplace, it's outrageously expensive, but if they cut the pufferfish wrong, it kills you. So it's kind of like, ooh, we're here, and a little whatever. I'm like, no, I'm good. I don't want raw fish. I don't care. I actually don't want the raw fish to have a chance to kill me. So lots of things going on. Okay, so we're going to talk about some other kind of individual issues with muscle fibers next. But in order to understand those, we need to talk about how we do muscle biopsies first. Okay? What you see here is a muscle biopsy. This needle that you see is called a Bergstrom biopsy needle. Okay? So we have the ability, in theory, to be able to do this here downstairs. We don't have Bergstrom biopsy needles. I don't believe we have microbiopsy needles. Megan, did you ever see anybody take a biopsy at Baylor? Yeah. Okay. I was going to say, Terry and those guys do them quite a bit. Yeah, that's part of why we haven't done them here in like 10 years, is there was some irregularity with our IRB paperwork and some things, and it didn't go great. But you may find this hard to believe, okay? But in the 1960s, when this was developed, this was one of the technological advances, along with the advent of molecular biochemistry, that revolutionized the entire field of exercise physiology, because it allowed us to learn things about an individual muscle in a person without having to kill that person. Okay? So the way this works is you make a little small, about maybe a little bit more than a centimeter incision. You insert the needle in. The needle is, it's not quite as big around as a pencil, but it is not a small needle. Okay? You insert the needle in. On one end, you have a syringe and a plunger. And so what you do is you apply suction, basically, to this. On the other end down here in the needle, there is this cutout window. And so you apply the suction, and it pulls a piece of muscle into this window. And then you mash the thing down, and it functions like a guillotine here. This is very, very sharp. And it cuts off this piece of muscle, and you capture a piece down in there. So you can see we've cut them open, we've stuck the needle in, we've applied the pressure, then we chop off our piece of muscle. You can also take biopsies of fat if you want to now, connective tissue. It just kind of depends. Then you pull the thing out, and you have a piece of muscle that looks like the eraser on your pencil. And when I was in Georgia, we would take those and we would mount them on an old popsicle stick, put them on a popsicle stick, put them in liquid nitrogen, freeze them, and then from there, now we have muscle from Kelvin that we can hang on to for forever if we want to. Okay? And so from that, then we can do different kinds of analyses. We can look at the individual muscle fibers. We can do muscle fiber typing. We can look at how much glycogen's in the muscles. We can look at how many mitochondria are in the muscle. We do a whole host of different things. And we can take a biopsy from Kelvin, and we can have him do bed rest for 90 days, and we come back and take a biopsy again, and we can see how things have changed, right? Are the individual muscle fibers bigger or smaller? Has the fiber type changed? Does he have more or less glycogen? Okay? This allowed us to do those things and not just look at the whole muscle. So people still do this, but a lot of what is done with this now, they don't even use the Bergstrom needles. They use these little micro biopsy needles. They just need enough tissue to be able to basically run like the coronavirus test. Okay? They do things they're looking for messenger RNA, they're looking for gene expressions. And so what we're looking at is there messenger RNA? Can we do something called real-time polymerase chain reaction? Take that RNA, make protein from it, and figure out, like, are certain genes getting turned on more or less at some time period after a bout of exercise, okay? If you've trained for 12 weeks, do you have more chitin in your muscle than you did before? Do you have more, whatever, do you have more enzymes from your mitochondria? So we don't always have to have as much tissue as we used to, because some of the stuff that you needed this for, very labor intensive as a person that had to be the person looking in the microscope and circling individual muscle fibers. very labor intensive, not super fun. We know a lot about fiber types and kind of individual sizes. Okay, so students in our department may ask, yes, in theory we could do this. We probably have to get a physician from the health science center to come down and take the muscle sample, but it's a thing that we could do. So we did a bunch of this in the lab that I was in when I was a PhD student. So it's not like the worst thing to have done to you. You get local anesthetic, we get people's out of the pain, you numb them up, and you cut them open. You don't always feel it when you get it the next day. Not great. The next day is not great. We would do things where we would like take one, and then they would come back to get a baseline. They would come back the next day. We'd take another one, and then we would have them do a bout of exercise with the incision still open, and then we would take it again out of the same incision at the sort of time points after the bout of exercise, because we were looking for gene expression in a kind of time course of things afterwards. I think at one point we did like 10 or 12 biopsies over the course of like a week in people. But to watch the people when your leg is sore, and I say sore, like, I mean, it really hurts, and you just have like an open wound basically to watch them. You come in there and say, it really is not a pleasant feeling. And then you'd have to like exercise that and like lift weights with that muscle, and people would just like cry. So.
How much did you pay him?
$100 a biopsy. So, and it's totally not coercion, right? So, also if we pay people $1,000 to not bear weight on their leg, we'll talk about that one. Okay. So the key thing that we learned from muscle biopsies, we learned about muscle fiber types. Okay. And so when I talk about fiber types, what I'm going to make a distinction of is most of the things that differentiate our individual muscle fibers have to do with the myosin and their mitochondria. Okay? So not all muscle fibers are created equal in that way. Okay? And so when we originally did this in the 60s, they thought there were, and they knew some of this from animal studies, they thought there were two kinds of muscle fibers. They called them slow twitch and fast twitch. Okay? And they knew this because in the animals, especially like rats and cats, you could look at the gastroc and you could look at the soleus, right? Soleus is very kind of endurance because it's a postural muscle, right? Dentist chair flashbacks, probably a weed eater, but the twitch portion of this comes from, they would take the muscle and they would anchor it on these two little pins, and then they would shock it, or they would bathe it in calcium or something, and it would contract, and they would look at how quickly it was able to turn on and generate force. And so if you imagine this, you've got a muscle and it's resting, then we turn it on, we give it an action potential, it generates force, and then it relaxes. Okay? Let's say this is a fast muscle. In a slow muscle, it may look something more like this. And so the amount of time it takes to go from nothing to the most amount of force is much shorter in a fast twitch muscle than it is in a slow twitch muscle. Okay? So one action potential, this is called a twitch. In theory, we can do this in my lab or Dr. Mike's lab, or even maybe now down at basketball, I don't know. If y'all have a chair and a stimulator. We did it before with Aaron's stuff. We can do this in your muscles, in a whole muscle, okay? And it will tell us, but they can pull out individual fibers. And so we knew there were some fibers that would generate force faster than others. And so there was this distinction between fast twitch and slow twitch. In general, fast twitch muscles are not good at long distances. They don't have a lot of mitochondria. They don't have a lot of capillaries that go to them. Their myosin ATPase is like a Hummer, okay? It's very fuel inefficient. It uses and wastes lots of ATP to generate its force. Conversely, these slow twitch fibers, they tend to have many more mitochondria, and they're more like a Prius, okay? They're very good at contracting over and over and over again, okay? So originally, we would do what we called histochemical, which is this old way of doing things. Now we can do what we call immunohistochemical, which is this new way of staining. So old histochemistry, which I unfortunately had to do, even though we could do this, we still did it this way. We incubate the individual muscle to biopsy with certain substrates and these different stains, and we're looking at the activity of the myosin ATPase enzyme. That's the place that we're looking, okay? The ability to come on and off, it's mostly, technically the ATPase is actually for off, okay? But the on and the off are controlled by how quickly the myosin ATPase can split the ATP apart and have the muscle relax, basically, okay? And so by doing all of that in different pHs with different substrates, different speeds of myosin ATPase will look different colors, and I'll show you all a picture of all of this, okay? So there is a fast myosin ATPase and there is a slow myosin ATPase. We could also look at metabolic enzymes, which would tell us about mitochondria. SDH stands for something called succinate dehydrogenase, which we will talk about. It's a Krebs cycle enzyme. We'll talk about that in metabolism. We could look for hexokinase, which is the, what, Kelvin? See, we'll keep back to Mike. We probably want you to know that. You better remember that. There's hexokinase.
It's the enzyme that kills from glucose to glucose.
That's right. So it's the first step in glycolysis. This is what traps glucose from the blood inside cells. You can look at that. LDH is lactate dehydrogenase. You can look at kind of how there's a couple of flavors of those LDH enzymes, if you will, that will also tell us some stuff that we'll get into when we do this. You can stain for how much glycogen is in a cell. There's some really fascinating old studies where they would take a biopsy, they'd load people up on carbs, see how much glycogen was in there, and then they would have them run or bike or do various things, lift weights, and they would do another biopsy, and they would look at how much glycogen had been depleted out of kind of those same muscles. There's some really, really interesting things that we know about where individual fibers that are innervated in part of the same motor unit are from glycogen depletion studies. So they would find like one axon, and they would artificially activate that one axon. And so you're getting a small amount of force, but it's the same muscle fibers. They're going to contract over and over and over and over and over again. They're all from one motor unit. And then you pull it out from the biopsy, and you see like the whole thing looks dark because it has all this glycogen. There's like these little individual, like 5 little fibers where there's just no glycogen. And so you're like, oh, those are all in the same motor units. You know some things based upon all of that. We can now look at lipids, and there's a whole bunch of stuff that we can do. Now we know, because of immunohistochemistry, we can actually look at the myosin heavy chain, which is going to have some other slight differences. And so what we know from immunohistochemistry, we changed our naming convention because of this. So they're no longer probably typically called fast and slow. They're called type 1, which is slow, okay? And then type 2, which is fast, but there are multiple types of type 2 fibers. And that's based upon the myosin heavy chain differences. The 2 primary ones that we see in humans are type A and type X, okay? Type X are the fastest, okay? It's got the fastest contraction time. They are absolutely like the least efficient. In you guys, that's going to be the ones you have the least amount of, okay? If you are active at all, you're not going to have very many type 2X fibers. Now, older guy like me, probably more sedentary, I've got more 2Xs. You put somebody on bed rest, and we're going to turn their type 2A fibers into type 2X fibers. They're going to have a lot more. The 2As, okay, their myosin ATPase is about the same speed, but 2A fibers, for whatever reason, tend to have way more they have way more mitochondria in them. They have more capillaries that go to them. They're not quite as inefficient, okay? So every one of you was born with some percentage of type 1 fibers to type 2 fibers. Thank or curse your parents. You have no control over it. We cannot change it unless you want to let me send you to space, or you want to let me give you a spinal cord injury, and you sit in a chair for years and years and years. Okay? no matter how much you train, no matter how many miles you run, you can't make your 2s into 1s. No matter how many sprints or whatever kettlebells that I'm doing down there, box jumps and thrusters and whatever CrossFit tells you, your 1s aren't becoming 2s, okay? There is an ability to shift 2A's to 2X's by being inactive, and to shift 2X's into 2A's by doing any kind of activity, running, walking, biking, weightlifting, anything, okay? That will become more important later on in class. We'll get into some more. Okay? Everybody with me so far? Now, within you, different muscles in your body are going to have slightly different percentages of kind of your 1s and 2s. Most of us are 60-40 one way or the other, kind of as our baseline. Your diaphragm is probably the slowest muscle in your body because it needs to be the most fatigue resistant. The fastest ones tend to be like these little tiny muscles that go to our eyes so that you can blink and keep shit out of your eyes really quickly. Okay? Most people are 60-40 one way or the other. Okay? We take stuff in your quad, that's where you're going to be. If you're 50-50 in your quad, you're probably 60-40 and you're slow in your soleus. That's probably the next slowest muscle besides your diaphragm. Okay? That's where we are. Something else that I want to say. Any questions? Anybody has questions about fiber types? Yes, please clarify.
So when you're saying that you can change them, they're not actually changing them, right? It's just the type 2A's are becoming more similar to type 1 and more similar to type 2X. Right?
They're still type, yeah. So this is where it gets complicated. So you will, with training, you will turn the heavy chain, okay, from 2X into 2A heavy chain. So there is an actual physical change that takes place that's going to be accompanied by a small change in the ATPase rate in that particular muscle fiber. When that happens, it also then seems to allow for the development of many, many mitochondria in those muscles. Now, whether it's an allowance or it's just that when you just need to be active enough to drive mitochondrial biogenesis, those two things may be linked. Whatever the signal is that drives the mitochondrial biogenesis may also drive the switch in the heavy chain, and vice versa. So they look still like type 2s, but they behave from a metabolic standpoint a little bit more so like type 1 fibers. Okay? Great elite distance athletes generally are well, 70, 80% or greater type 1 fibers. Okay? Great sprinters generally are going to be 80, 90% type 2 fibers. When you're thinking Olympian, even like really, really good collegiate folks, you're probably going to be on the tails of that normal distribution on both sides. Okay? The rest of us regular schmoes are somewhere in the middle, in the midst of all of this. 2s tend to be able to generate a little bit more force than 1s. There's some debate as to which ones have a greater capacity to hypertrophy and increase their size. There's some evidence that 2s can grow more than 1s. I'm skeptical of that. You guys will see that when you do like normal contractions, anything when you walk around and you do low force contractions, you're always using your type 1 fibers first. They're always being active. And so it may just be that when we find people to train, their 1s are already trained. Their 1s are already as big as they're going to be. So the 2s are the ones that grow. Not because the 1s can't grow. It's just that they already have. So there's some, it's hard for us to do fiber type, well-controlled kind of reductionist experiments in that way. So it gets a little bit complicated. I do also want to mention, because it will come up later on, I lied when I said that the 2s come in only the 2 flavors, the A's and the X's. There are actually what we would call hybrid fibers. There's things that exist as a spectrum in between those, okay? And then there are also things that are what are called type 2B fibers. We used to in humans, if you read old human papers from the 60s and the 70s and even in the 80s, you will read about type 2B fibers in humans, because those fibers look like fibers from these animal models, okay? We now know when we can do the immunohistochemistry that we do occasionally have 2B fibers, but most human fibers are 2Xs. The 2Bs are going to become, they're even faster and even less efficient than the 2Xs. You typically will see those in people that are like bed rested or bed in space, where they're like super duper duper sedentary, okay? But it is there. And there's these hybrids that exist on some spectrum in between all of those things. I'll show you guys some data from some resistance training studies at the end of class where you'll see what happens to the hybrid fibers. Basically, when you become more active, your hybrid fibers all go away, right? So it's kind of an interesting thing. Okay. That's what they look like, fiber typing, blah, blah, blah. This is what, if you look at things under a microscope, so this would be what you would see from one muscle biopsy. And so you take the biopsy, and I know this because I messed up multiple blades doing this, but so you have the biopsy, which is here, and you assume that all of the fibers are mounted running kind of parallel like this. And we put it on in something called a cryostat, and it's basically like the sharpest razor blade you've ever seen in your life. And you put it on there, and it's like a deli meat slicer. You put the thing in, crank it, and it chops off these little tiny, like you can see through them, like tissue paper thin slices of the muscle. And you mount that on like on a slide. And you take another slice, and you mount that on a slide. And the third slice, and you mount that on a slide. So what you have then is these serial sections. And so you would take one and stain it for the ATPase. So this is going to be myosin ATPase function. And so what you can see in this particular one, the white fibers here, those are slow twitch. The black ones here, there are going to be 2As, and then the gray stained ones are going to be 2X fibers, okay? Then you would take the next section, which has exactly these same fibers in exactly the same location. And you might stain for succinate dehydrogenase. These are not serial sections here, okay? But I couldn't find a good picture. I don't know why I don't have any, but the darker this is, the more mitochondria there are. So what you can do then is, you know, here's my fiber type. You can circle each individual fiber to know how big it is. Then you can look at what's the signal intensity for SDH, what's the signal intensity of the same ones for glycogen. You can match all of those things up, and that's how we would know how much glycogen do we have, what's going on, how many mitochondria do we have. So now we can do that just by pulling out a tiny piece of muscle. Talk about those, talk about that. Now we'll come back to all of that. All right, last thing, then I'll let you guys go. What determines fiber type? I told you guys it's genetic, okay? Also a little more complicated than that, okay? There are genes that control what you're going to have. The interesting thing, though, is that associated with each fiber type, the fibers that are of a particular fiber type, all slow twitch fibers are going to be connected to the same collection of motor neurons. So within a motor unit, all of the muscle fibers that are connected to the same motor neuron are all going to be of the same fiber, the same fiber type. There's some fascinating studies where if I cut the axon, right? We take poor Keldon, I cut the axon going to some of his quad muscles, right? And then I swap them around. I take an axon from a fast twitch motor unit, and I connect it to fibers that were in slow twitch motor unit, and vice versa. Then we can flip the expression of what's happening in those muscle fibers. So there is a school of thought that it's actually the neuron and the nervous system that controls something about having, and it's not even physically connected, it's just that something is crossing the neuromuscular junction, okay? That activity is driving the expression of those particular genes in those particular muscle fibers. So we can switch them. In old people, type 2 or motor neurons that connect to type 2 fibers tend to be more susceptible to the accumulation of neurotoxin. So over time, grandma is slow for a reason. Some of her motor neurons that go to type 2 fibers have died. And axons coming from her type 1, that are normally going to type 1 fibers, will sprout off and try to go and reconnect to those fibers. Because if the neuron dies and the connection dies, the muscle fibers will die. The muscle fibers will go away. So you will get this, we call it collateral sprouting, where they will want to try to go over. Existing functional ones will try to branch off and grab those muscle fibers that have lost their connection. That happens, and you turn the 2X fibers, the 2A fibers have now become type 1 fibers. So there's some fascinating aging things where old people have very, very slow muscles, okay? And they have these, sometimes they have these really big motor units, and so they do really herky-jerky movements, and they do them slow, which is why grandma falls down, or why she can't drive. Okay? All right.
Could you keep the motor neurons from dying off just from training through like even like an intervention of like velocity-based training, I think?
So there is some evidence that people that are more active called inducing to have less motor neuron death. How much of that is simply due to we know that activity and signaling through the nervous system to the muscle fibers is good for the health of the neurons and the axons and the neuromuscular junction and the muscle fibers. How much of that is also don't eat fish that has mercury in it, or some of those kinds of things. It's probably some collection of all of that. I don't really know that I want grandma going to CrossFit and like doing velocity training, but we do know that old muscle tends to respond to training just up until maybe the 80s about as well as young muscle does. And if you can safely do it, the way that you learn to do things fast is you have to practice them going fast, right? And you have to preserve your fast twitch muscles. So yes, you're to partially answer your question, yes, we can. That would probably be a useful thing. I say grandma, but I should say I don't know that I want my mom doing. I don't know that I need my 72-year-old mother doing like depth jumps and doing bench press with 25% of an RM going as fast as she can, but it would probably not be a terrible thing. Right? So,
Dr. Black,
you
is
say
there,
velocity training, and I think of I don't just think like in an older person, velocity training is, hey, can you go jog, or can you do something with just any amount of speed? I'm thinking of like we've got athletes like really, really trying to go fast.
Right.
Or the bros in the weight room who have no idea what they're doing, like bench pressing and like smashing it off of their chest, just like because they saw a website that's what somebody does. Right? Okay. All right. We will call it quicksies here.
wait,
We
Dr.
got through this. All right.
Black,
So let me pull up.
can I ask a question real quick?
Yes, of course.
Is there any advantage of having type 2X over type 2A? Like, can type 2X generate more force, or is it just better to have type 2A?
Hold on, Kelly. I'm going to need you to ask that. I'm going to try to turn up the volume. I'm going to have you ask that again because it's hard to hear. Okay, try again, please.
Is there any advantage of having type 2X over type 2A?
Sure, you can be Usain Bolt. Other than that, no. And So there's a school of thought, or there was at one time a school of thought where people would train sprinters and they would train them up a bunch, and then they would actually have them like not do anything for 3 weeks or so, or a month before their competition. I don't know why I'm looking at the projector, because that's where your voice is coming from, guys. But they would do that in an effort to try to get a little bit of fiber type conversion from A back to X. What we know now is that if you don't do anything for like 3 weeks, that you lose some of the nervous system adaptations that allow you to run with proper form and some of that. But generally, no. And we know that because if you do anything, if you are active at all, you're going to mostly start turning your X fibers into A's. And so there doesn't seem to be a lot of adaptive advantage.
You can't like generate more force with X as compared to
So this is the graduate class, it depends. Did I tell you guys, I told you guys that the answer to every question I ask you could always be, it depends, okay? So there's some data that are suggestive that 2X fibers can generate more force than 2As. And then 2As can generate more force than 1s. However, part of that may be because those fibers tend to be a little bit bigger, okay? And so if you actually look at them on a per size basis, if you normalize for size, there's some evidence that maybe they all generate a roughly equal amount of force. But the other part of that is, well, 2s are just bigger. If you have more 2s, then are you just going to be bigger? Maybe. So it's complicated, Cal. So maybe in some scenario you could, if you had those, generate more force. The problem is, to have a bunch of those, you have to be really, really inactive, and that is not congruent with doing training to have the nervous system adaptations to allow you to generate large amounts of force. So that's a good question, though. That would be a good, like, you know, come up with a scenario on the take-home test. Like, come up with a scenario and explain to me why it would be of some sort of advantage to have lots of 2X fibers. I don't know, like I'm King George and I just sit there. And I need to be fast to do like one thing. People just bring me stuff. Because, you know, you got a bunch of X's, you do like 2 contractions and you're tired or you're fatigued. So, also not a great adaptive advantage for finding food when you're older. So, okay. Anything else? All right. Then for Tuesday, okay, for Tuesday, we'll do critical thinking stuff over some of this on Tuesday. We'll probably start the control of force production stuff on Tuesday. No, I'm sorry, God, this is Tuesday. Thursday, we'll probably have our next quiz, though, next Tuesday. All right. So we may start some stuff, but you don't have to have your critical thinking questions in that set of uninterjective stuff. Cool. Okay. All right. Then, ladies, I'm ending the Zoom session. Okay.
Okay, thank you so much.
You got it.